Okay. Well, Dr. Oder and I have been speaking. I guess I have to give him up at this point and let him give his lecture, but there's so many interesting things to talk about. So welcome, everyone, to the DCRI Research Conference. Our speaker today is Dr. David Oda. David is a principal investigator and group co-chair of the American College of Surgeons Oncology Group, as many of you know. It's a multi-center cooperative group which is funded by the National Cancer Institute. He's also professor of surgery at the Duke University School of Medicine, where the ACOSOG administrative headquarters is based. Dr. Oda's research interests have focused on the biology of breast, colorectal, and lung cancer. He has a strong interest in improving the standard of cancer care through prospective clinical trials, and we're fortunate to have him here to do that. He's authored over 145 papers and a member of several scientific and surgical organizations. And today's topic is mutational analysis and clinical trial design in cancer therapeutics. So, David, thank you for joining us.
All right. Thank you. It's been a while since I've given a presentation to the DCRI on where we're at with cancer therapeutic trials within ACOSOG. The American College of Surgeons Oncology Group is a multi-site clinical trials group that's funded by the National Cancer Institute. And so we conduct clinical trials. Some of them are surgically oriented, but some of them also have a strong attachment to medical oncology. And I think this is of what's interest to many people here at the DCRI, but also campus-wide. So what I'd like to do is just give you a bit of a update as to what this organization has been doing in the last 2 years, 3 years, some of our accomplishments, and where we might be going in the future. So one of the things that's becoming more and more apparent in oncology, and I suspect that this is going to happen in other fields as well, other areas of medicine, is that we're starting to see mutations of specific genes in cancers. And some of these mutations are very important because they are not only are the cause of the cell proliferation, but also become the targets for inhibitors of these very specific pathways. And so I'll try to give you some examples of this. And some of these mutations of key proliferative pathways, growing pathways that stimulate growth. They could be amplified genes, there could be deletions, there could be insertions, point mutations, and things like inversions. So they're starting to see through a lot of basic science research that a lot of these stimulate or cause an activation of the proliferative pathway. And that proliferative pathway actually starts to become the driver. It starts to drive the growth of that particular cancer. And so this allows medicinal chemistry, it allows industry to start to develop very specific targeted agents. And so these mutations seem to be very important. They lead to what's called constitutive activation. In other words, they're turned on all the time. And they cause the tumor to grow. But because they are constitutively activated, they are driving the growth of the cancer. And so it becomes a potential target for new therapeutics. So the classic examples of this include what's called anti-endocrine therapy. In breast cancer, the cancer, about 70% of the breast cancers are stimulated by estrogens in the body. and these are called estrogen-positive breast cancers. And this has been known for about 70 years. A guy by the name of Charles Huggins at the University of Chicago discovered this for prostate cancer, and it eventually became applied to breast cancer as well. Now, what breast cancer and prostate cancer have in common is that they are hormonally stimulated cancers. And in the case of prostate cancer, it's androgens, which are male hormones, and in breast cancer, it's estrogens. And so anti-endocrine therapy has been sort of a mainstay of breast cancer treatment for decades now. There's another classic pathway, and that's a specific growth pathway called HER2/neu. It's part of the EGFR family of genes, and when this particular gene is overexpressed on the surface of the cancer cell, in other words, it becomes amplified, it becomes a target for therapy. And so there's a very famous breast cancer drug called Herceptin, which blocks the HER2/neu pathway, and in about 25% of breast cancers, this drug has just revolutionized the treatment of breast cancer that is HER2/neu overexpressed. One of the interesting things that has been recently discovered is that HER2/neu is not only overexpressed in breast cancer, but it's overexpressed in ovarian cancer, esophagogastric adenocarcinoma, about 10 to 15% of cases, and endometrial cancers. So what this is saying is that it's not just these types of, one kind of cancer, one specific cancer uses this pathway, but other types of cancers are using that same pathway. Not all the time, but as in this case, one is 25%, the other one is 15% for esophagogastric adenocarcinomas. And what that has led to is that there is a clinical trial that has started for the esophagogastric adenocarcinomas using Herceptin as the treatment. And they're screening those. There's a way to screen and identify HER2/neu overexpression with a FISH assay. So you can actually start to subtype these particular cancers based on a biomarker. And so that trial is underway. So now the problem that we're seeing in cancer is that there are many proliferative pathways. There's the EGFR pathway, there's KIT, there's BCR-ABL, the list goes on and on and on. There are probably about 40 or 50 known proliferative pathways in human biology. And the question is, when you see a mutation, which one of these becomes the driver pathway of cell growth, as opposed to being just a passive or a passenger pathway? And I'll give you some examples of that. But what's being discovered is that there's a mutation which converts this particular pathway all of a sudden into a driver, and it drives the growth of the cancer. Now, one of the things about these types of pathways is that there is a kinase region internally in a lot of these pathways, internal to the cell membrane. And a kinase is an enzyme. And when you start dealing with enzymes, and this is where it triggers the growth, enzymes can be inhibited. And so you can start to develop what are called small molecule inhibitors of that particular pathway. So let me give you an example. This is a clinical trial that ACOSOG ran. This is a phase 3 clinical trial, double-blinded study of adjuvant imatinib versus a placebo in patients who had a resected primary gastrointestinal stromal tumor, GIST, rare disease. You know, only about 5,000 new cases a year in this country. But it was one of the first to develop a biomarker, and that biomarker was overexpression of KIT. KIT is a pathway, proliferative pathway, and in gastrointestinal stromal tumors, KIT is overexpressed. And so we ran this trial comparing placebo versus adjuvant imatinib, and then followed the patients. And what we saw was that if you received 1 year of the which one is the pointer? Does anybody know? This right here. Just push there. No, that's okay. All right. So what this is the survival pathway. The lower curve is the placebo arm, and the upper curve is the imatinib arm. And you can see that there's a separation in the curve, and that this led to the eventual approval of this drug as adjuvant use for gastrointestinal stromal tumor, and DCRI was very much a part of helping Novartis file for the FDA and getting the tables ready in preparation for the FDA registration. Now, the patients received the drug for only 12 months. And so you can see at 12 months there's a separation. But the protocol was designed for only 1 year of therapy. And so you could see in the placebo arm there was a certain relapse rate. And in the imatinib arm, which is on the top level, they started to relapse, say, at around between 18 and 24 months. And that's because the drug was only given for 1 year. But it makes you wonder, you know, should the drug be given a little bit longer? Obviously, 1 year is not enough, and those kinds of trials are ongoing. So this is kind of a classic oncology adjuvant trial. You have a placebo, you have a drug, you try to prevent recurrence, and you see the separation in the curves. And when they separate long, you know, or far enough, hazard ratio here was 0.35. P-value was less than 1 in 10,000. So actually, the trial was stopped early by the Data Safety Monitoring Board because the P-value and the hazard ratio were so strong that they stopped the trial early because the significance was so great, and it wasn't ethical to continue on enrolling patients into a placebo trial. This is the kind of activity you see with this drug imatinib. This is a patient in that upper left corner is a patient with a rectal GIST. You can see a large mass in the pelvis right here. The rectum is actually right here. It's all been pushed to the side. Huge tumor. Patient had problems with bowel control, had trouble with urination. The bladder had no capacity. This thing occupied the pelvis. And the patient was started on imatinib because we had a tissue diagnosis of GIST, and we confirmed that with IHC, which showed overexpression of the KIT pathway. At about 2 weeks, the patient came back and said, you know, I don't have any more diarrhea, and I'm able to urinate normally. 2 weeks of treatment. At 8 weeks, we repeated the CT scan, and you could see that the tumor is much smaller. This is the rectum right here, and this is the bladder right here. So it was a very strong response. And this is normally what oncologists will show you. They'll always show you the good pictures and the good cases, right? You know, happy ending, you know, symptoms go away. But there's, so this is the good side to the story. But like any story, there's always a dark side. And that dark side is that not all the patients respond this way. So why is that? Well, in this particular case, the study PI had a grant from the NCI to look at the primary tumors that were resected. And what he started doing was he looked for mutations of KIT and tried to figure out, is there some relationship of a specific mutation in KIT and the recurrence rate. And what he found was that there was a very specific mutation called exon 11 deletion. About 93 of the patients had this. And you could see the relapse rate here in this green line. This is in the placebo arm. And you could see that they were relapsing a lot faster. And this is a frequently occurring mutation, exon 11. And almost about, it makes up a significant, exon 11 mutations make up a significant number of these mutations found in KIT. What's interesting is that the KIT can also be known as wild type. In other words, wild type means that it's normal. There's no mutation that's detectable in that particular gene. And you can see that there's a big separation here between the wild type and the exon 11 mutation in the placebo arm. So if you look carefully, though, at the data, at the Z9000 data, what was found is that if you look at the exon 11 mutant cases for the recurrence-free survival, you could see that this is the placebo arm right here, and this is the treatment arm right here, and this is the 1 year of therapy. And you can see that the response is even greater. And that the wild type, you could see in this, here, there was no difference in survival. So somehow that exon 11 mutation sets up these tumors to be more responsive to imatinib. And this is what's been found not only in an adjuvant setting, but in a metastatic setting as well. So the data is starting to show that there is a specific mutational type, not just overexpression. Now, one of the things about KIT exon 11 mutation is that it occurs in about 67% of GIST cases. It's very common. And so when you look at that survival curve that I showed earlier of imatinib versus placebo, that separation was mainly due to the exon 11 mutation in 67% of the patients. Now that's really important because if the mutation, the activating mutation, occurs in only 10 or 15% of the population, and you do this big generalized type of adjuvant trial, you're not going to show any benefit. Okay? And so mutations seem to be very, very important. Novartis, which is the pharmaceutical company that makes imatinib, was just lucky that 67% of the mutations tended to favor the treatment. And so, you know, they were very happy, went off to the FDA, and got approval. But some of it was dumb luck. Okay? And I'll show you, I'll tell you another story about another company that spent, billions of dollars, $1 billion running a clinical trial, chasing a pathway with a targeted agent in which the key mutation was only 10-15% of the population. Guess what happened to their trial? Well, didn't show any benefit. All right. So in this case, we were just very lucky that it was 67%. So now mutations aren't the only thing that are important. It gets a little bit more complicated because you might say, well, geez, maybe we just need to do genetic analyses of the tumor and we have all our answers. And for those of us who are physicians, it's not so simple as that, because there are clinical pathologic factors that also add into this as well. And so one thing about GIST is that if the primary tumor is in the small intestine, there's a higher likelihood for recurrence. And this is a multivariate analysis. If the tumor's bigger tumors mean more likely of spreading, and that's also a factor as well. And then there's also the pathologists like to do this. They like to count mitoses. Mitoses mean cells dividing, and they look under a microscope, and they can actually count to see mitotic figures in the field of cancer, and that also seems to be significant as well as the exon 11 mutation, which is seen here. So one of the things that is being considered here is a follow-up study trial in which we take into consideration tumor location, tumor size, mitotic rate, as well as the genotype. So there's it's not just genetics, but there are other components, there are other parameters that seem to influence recurrence here. So this would be a means to stratify these patients and select the high-risk patient population. So we still have to be doctors. We still have to know what the small intestine is. We still have to know what tumor size is, know how to read a path report, and have our pathologists looking at the microscope, looking at the cells under high power. Now, the exon 11 story, mutation story, goes on further because not only is it found in GIST, but exon 11 has also been found in a rare type of melanoma called acromelanoma. It's the melanoma that occurs on the hands, the palms of the hands, and the soles of the feet. And these tend to be very aggressive melanomas. They're very late stage in diagnosis. And there's also another type of melanoma that's called mucous melanoma that occurs in the mouth. It occurs in the anal canal. And these are also very aggressive melanomas. And in fact, anal canal melanoma is by just the location unresectable at the time of diagnosis. There's no point in doing any surgery to excise it because the mortality rate is so high. But what they found is that this exon 11 KIT mutation in some of these melanomas, and it's not like the skin, the cutaneous melanomas that we see. It's on the palms of the acral and the mucous melanomas. Now you could look at melanoma in another way, because what has been discovered in melanoma is that there is a key mutation in a pathway called BRAF, and about 50% of melanomas express have a BRAF mutation. And there's actually an agent now that is targeting this particular mutational pathway. And there was a recent article, I think, in the last couple of weeks in the New York Times about this, about the ethics of a placebo versus BRAF inhibitor trial. And the people who were randomized to the BRAF inhibitor were having spectacular responses, and the placebo arm, they were dying. And the primary endpoint of the trial is overall survival. So that means you let the people go and die. And so there was lots of ethics about it, because this drug is working extremely well if there's a BRAF mutation. So this also starts to impact how you approach the FDA and how you design these trials, because an overall survival endpoint means you're waiting for people to die. And that may not be the best way to design the trial. You know, the primary endpoint could be like progression-free survival, as opposed to letting people die, and that allows people to cross over onto the other treatment arm that seems to be effective. But melanoma also has this, some of them have the KIT exon 11 mutation as well. And one of the things that's being discovered is that these mutations are mutually exclusive. And what that means is that if you have a BRAF mutation, you're unlikely to have the KIT exon 11 mutation. And if you have the exon 11 KIT mutation, you're unlikely to have the BRAF mutation. So there's this mutual exclusivity story that's starting to come out. And I'm going to tell you another story where that's happening as well in lung adenocarcinoma. So you can start to see a little bit of pattern here that's being developed. Now, the American College of Surgeons Oncology Group is taking this a step further with breast cancer and applying a whole new technology called whole genome sequencing. And we're doing this within a trial setting. This is ACOSOG 1031. It's a trial in which you have women who have highly ER-positive breast cancer, clinical stage 2 and 3 disease. That means they tend to have larger tumors. A breast cancer that's 5 centimeters or greater in diameter is by definition stage 3 disease. They don't have metastatic disease, but they have what's called locally advanced disease. And those kind of cancers, breast cancers, normally require, if surgery is done first, require a complete mastectomy. In other words, entire removal of the breast. Now, one of the things that is happening in breast cancer treatment is instead of going to surgery first, why not treat them with a drug and reduce the size of the cancer? And this is a trial in which it's actually more of a phase 2, not actually a phase 3, but patients are equally distributed to 3 different aromatase inhibitors, exemestane, letrozole, and anastrozole, and they're all called aromatase inhibitors. And the patients go on to the trial. They get treated for 16 weeks. Cancer shrink. Not all of them. Most of them do. Most of them shrink, and surgery is then performed. And one of the things breast surgeons like to do is instead of doing a mastectomy, why not just perform a lumpectomy, especially if the cancer shrinks. And so this trial was actually completed, finished accrual, and the clinical responses were assessed during the trial. This was the primary endpoint using caliper measurements. So you could actually feel these tumors, and you could actually feel them shrink down. And so the criteria for response was based on the WHO criteria, and the complete response, which means you couldn't feel the tumor anymore. It shrunk to the point where you couldn't feel it. It was still there, but you just couldn't feel it. Or there was a partial response. And so out of the 377 patients, 249 had a response. So that's great. Good news. Cancer shrinks, and I, as a surgeon, I get to take the patient to the operating room. Instead of doing a mastectomy, I can just do a lumpectomy, and the breast is preserved. So that's pretty good. Great. But the dark side of cancer treatment is this right here, and that is 82 of the patients had stable disease or they actually progressed. And that's the conundrum we face in cancer is, you know, why did these patients here, why didn't they respond? These are all highly ER-positive breast cancer patients receiving anti-estrogen therapy. Not all of them respond. And this shows you some of the results of surgery. This is the surgical outcome of the trial. Before the patients went on treatment, the surgeons had to determine whether the patients were marginal candidates for lumpectomy, which is referred to here as BCS, breast conserving surgery, or whether the patients required mastectomy. So they the surgeons had to commit to this up front. And you can see that the conversion rate, if you were in that mastectomy group, the conversion rate was 51% to breast-conserving surgery. That's pretty good. That's pretty good, all right? But a lot of patients still went on to mastectomy. Now, the surgeon sort of, this is one of the clinical reasons to do the trial. Now, are there some laboratory reasons or scientific reasons to do this trial? And one of the things that you do in this kind of setting where you have neoadjuvant therapy before surgery is that you have one tumor, and surgeons, we're pretty good at getting tissue. We like to poke around. And one of the things that you can do in a trial setting like this is the surgeons can get a piece of that tissue, the tumor tissue, with a biopsy needle. And so before the patients go on the aromatase inhibitor trial, we can get a piece of that tissue and we can bank it. We can bank it, and then we can look for the response. So we have what's called a phenotype. We have responders and non-responders in a clinical trial setting like this, and we have freshly preserved tissue. And that tissue, with the appropriate consent, you can do things like genetics and whole genome sequencing. Dr. Elaine Martis is at WashU and chairs our Basic Science Committee and runs the Genome Center at WashU. This facility here at WashU has 50 whole genome sequencers. They got a whole building full of these genome sequencers. Now, the human genome was first sequenced about 10 years ago. It took them about 4 years to sequence one human genome. Probably cost us about $4, $5, $6 billion of our tax dollars to sequence that genome. That's one genome, 4 years, $6 billion. With these new sequencers now, and this actually is just, this is just one sequencer. You can see another one over here. They got 50 of these things. And they can sequence the whole genome now in 7 days for about $50,000. And so that's a tremendous drop in price and great increase in speed. And about every 2 years, they come up with a new generation of these sequencers. And these sequencers are nothing but microchip processors. They're all robotic. They're microprocessors, and they're software. And so the next, so every generation of sequencers cuts the cost in half and cuts the time in half as well. So in about 4 years, these sequencers will be able to do whole genome sequencing probably in maybe 2 days, and at a cost probably around $10,000. So it's like the PCs on your desktops. It's the same sort of thing. And so these machines are going to get faster and faster and cheaper to do. So it's probably, we're going to see this not only for cancer, but this is also be applied at WashU for other diseases, such as neuropsychiatric diseases, infectious diseases. So there's a lot of activity going on with whole genome sequencing. The problem now comes, what do you do with this amount of data? Because you start to generate tremendous amounts of data. And so what we did was we looked at this particular technology for this particular trial, and one of the things that we did was we got a core biopsy before the patients underwent treatment. And that biopsy was preserved. It was frozen and sent to our tumor bank where the DNA was extracted for whole genome sequencing. So one of the things that you have to do when you do this kind of genetic analysis is you've got to have a phenotype. So if you're going to look at genes and mutations, you want a phenotype. And a phenotype is what you see, what you clinically see. So a guy like a monk by the name of Gregor Mendel was looking at red flowers and white flowers. Those were the phenotypes. Now, physician, what we do is we look for a clinical outcome as a phenotype. And so in this case, we took 25 of the hormone responsive cases and 25 of the hormone unresponsive tumors and performed whole genome sequencing on the tumors. So we had 50 genomes or 50 cases, and you have to have pairs. You have to have not only the cancer DNA, but you also have to have the germline DNA. And the germline DNA is your DNA that you get from your white cells. So these patients had consented to this. The white cells were obtained before treatment. The tumor was obtained. DNA was extracted. And so you could then begin to do whole genome sequencing. The problem is this. You know, if you look at 50 cases, and each case the human genome is about 3 billion nucleotides, 3 billion bases. And you start doing a little math, you're going to end up with 300 billion bases of information. That's a lot of data. And this is just on the 50 cases. So we've got other types of trials in which we're doing whole genome sequencing. So one of the problems with these sequencers is storing all of that data. And so they have monstrous amounts. They have petabyte type of storage capacity at the WashU Genome Center. So it's a tremendous amount of storage capacity, all this data being generated. The problem is this. How do you analyze all this? How do you analyze 300 billion bases? And that's highly problematic. And hopefully, you know, we have some mathematicians or search engine software or something to help decipher this. Because that's probably where the field is lagging. Because as I said, the sequencers technology is moving a lot faster. But one of the things that they have done in looking at some of the sequence data is they looked at a specific pathway called PIK3. And they've looked at 38 of the 50 samples, and they've found point mutations, 12 in the responsive patients, and 7 in the resistant patients. And what they found is all 7 of the aromatase inhibitor resistant patients, their particular tumors have a mutation in exon 20, which is the kinase domain of PIK3. Now that's kind of interesting because there are PIK3 inhibitors that are in development right now. And this kinase domain is where these activated mutations occur. And so there's a great deal of interest in then tying a therapeutic trial, PIK3 mutations, for these aromatase inhibitor patients. And this may be the driver pathway of these aromatase inhibitor resistant patients. So there's a bit of discovery going on here. Now, this is not the only story. There's an EGFR receptor, which is very specific in lung cancer, and it also has a kinase region as well. And there are tyrosine kinase inhibitors. One is gefitinib and one is erlotinib. And they're very much like imatinib is for KIT. These are specific inhibitors of the epidermal growth factor receptor. And they've been developed, probably been around now for about 20 years. The story about that pharmaceutical company that tried to do an adjuvant trial for gefitinib, AstraZeneca, treated all lung cancer patients with this drug. And it turns out that this drug only works if there's a specific mutation in the kinase region. And this only occurs in about 15% of lung adenocarcinomas have this specific kinase mutation inhibitor. So if you treated all of lung adenocarcinomas with gefitinib, what you're going to find is no difference in survival. because the number of, the percentage of kinase mutations is only 15%. And this is actually the population that you want to study. This is the subset. And so we're actually moving toward a trial design within ACOSOG where we take patients who have resectable lung adenocarcinoma, and the surgeons have to get the patients consented before surgery. They obtain the tissue, and the tissue is then sent off for mutational analysis for EGFR. And those that are have the EGFR mutation can then be put into this randomization scheme where you have EGFR TKI right here versus placebo. So there has to be a great deal of screening that goes on here by the thoracic surgeons who see this disease, and they have to have patient consent. So we're in discussion with, and we're developing this trial. Now, how do you screen for 2,500 cases to get 378 EGFR mutations? Well, you got to partner with a diagnostic company. And so we're partnering with a diagnostic company to do the screening of 2,500 cases so that we can find 378 EGFR mutations. And that's how we're trying to get around and develop this particular trial design. Now, what's actually very interesting is that there's another pathway called ALK, A-L-K. And ALK occurs in about 9% of lung adenocarcinomas. And what's been discovered is that this ALK mutation, or it's called a fusion, is mutually exclusive of the EGFR mutation. So that if you have the EGFR mutation, you don't have the ALK mutation. And the same goes for ALK mutations. If you have that, then you don't have the EGFR mutation. So you could actually you have another subset of patients that you could potentially include into an adjuvant trial. And it turns out there's an inhibitor for ALK mutations. It's incredibly effective for lung adenocarcinomas. They get spectacular responses, sort of like that picture I showed you with the GIST tumor in the rectum. They get rapid shrinkage of these tumors. And we're trying to negotiate with that sponsor for the ALK fusion inhibitor to try to include it in this trial design, because the biology shows that these mutations are mutually exclusive. So, but that leaves us with, what about the other 75% of lung adenocarcinomas? And what we're incorporating into the trial is whole genome sequencing, because we're looking for other specific mutations and other proliferative pathways that may have an inhibitor or a monoclonal antibody in the pipeline. So ACOSOG is a group of, is a multi-site clinical trials organization that has surgeons. And one of the things surgeons have access to is this fresh tissue and the ability to consent patients before they take them to the operating room. So we're using this as a key strategy in the development of our trials. And we're continuing to do these types of adjuvant, neoadjuvant trial designs that need integral biomarkers, targeted agents, and one of the things that we're struggling with, and this may be a common phenomena that's being seen by other disease groups here at DCRI, and that is you have a therapeutic agent, but you also have an integral marker. So when you do the trial, what the FDA is looking at is not only the therapeutic agent, but they're also looking at the integral biomarker as well. And in oncology, we're struggling with the FDA on this, of putting together a trial design like we're doing for the EGFR. Now, the EGFR mutation trial is not going to be an FDA registration trial. And the reason is the patent is going to expire here in the near future. So the company's not interested in an FDA registration type of activity. They are interested in practice-changing type of data, which is a publication, as opposed to a labeling type of registration, and so or labeling change. So we're struggling a little bit with the FDA on this. I think a lot of the sponsors are struggling as well, and I'm not sure. I suspect that other diseases here at the DCRI, as you start moving toward these integral biomarkers, may be confronting that same situation as well, and be very interested in hearing about that. The interesting thing that we're doing with this whole genome sequencing is that we're starting to see massive amounts of data, and we don't seem to have quite the software packages to look and analyze that data in a timely way. It's like going from one gene at a time, and it's a very slow process. And somewhere in the future, there's got to be some sort of software search engines like Google uses that you see, sonar algorithms. It's another mathematical theory that could be applied. Trying to look at this massive amounts of data. And I actually have an appointment, a luncheon with the Duke University Center for Theoretical Science and Mathematics this week to talk about, how does an MD, interact with mathematicians, software engineers to try to come up with this kind of software that will help us analyze this data, because I think this is a huge problem that we're going to be seeing in biology in the near future as these sequencers become more common. And I think, it's becoming a lot cheaper. And so the technology of sequencing and our understanding of diseases becomes greater, especially as MDs. But somewhere there's got to be that software that helps analyze that data. And I don't see MDs coming up with that kind of solution. We're not wired to do that. We don't write software. We're not mathematicians. And probably explains why a lot of us went into human biology and medicine as opposed to why some people go into mathematics and statistics. So thank you very much. Be happy to answer any questions.
Thanks, David. It's very, very interesting. I have a question in the back, Danny.
So two items. The first is one of the things that we're starting to find out is that when we hold the IND for companies for regulatory approval for their therapeutics post-licensure, so they are trying to either expand their label or they are going for something like exclusivity, or they are going into the European Union. We have found that the first thing out of the words of senior management from a couple of those companies is, I can't believe they are letting you guys do X, Y, and Z. And from the agency side, we have been told on more than one occasion, we are letting you do X, Y, and Z because you are not the pharmaceutical company, and we do not believe that you have the conflict of interest to the level that the pharmaceutical company does. And we know that we're going to have access to these data in a way that you guys are not going to shade the data. Mm-hmm. We've been told that on the record. So that's one thing. When you're negotiating with these trial designs, one of the things that we found on ways to improve trial designs has been for us to hold the IND, and it's allowed us to move trial design forward. I have no idea, given the division that you're in front of, whether that is helpful advice at all.
All right.
My question for you is, when you talk about survival versus progression-free survival, can you give an idea of the numerator and the denominator by which kind of progression-free survival has been later repudiated as a primary endpoint relative to survival?
Right. So that's one of the problems with progression-free survival, especially if you deal with metastatic disease, is that with metastases, you can have 5 or 6 metastases in your body. Sometimes you could have 10. And what happens is when you start patients on treatment, maybe 5 of the 10 lesions start to respond, and the other 5 don't respond. And so, which was the index lesion that you chose at the beginning of the trial? And, none of the index lesions were, there were no index lesions. And so if half of them responded and the other half progressed, is that, progression or is that response? And so it gets very cloudy. So I would, I think that's one of the problems with progression-free survival in a trial design as a primary endpoint in oncology is that you can have responses, but it may not ultimately lead to overall survival. And so that's why they're sticking to their guns with this BRAF inhibitor trial in melanoma. But it's pretty painful to watch people, progressing along and just, and watching them, because there's no other treatment. There's no other alternative. Chemotherapy is nothing. And so, the story in the New York Times is that there were two cousins, and both with metastatic melanoma, both enrolled in the trial, and one got the experimental drug, and the other one got the placebo, and they, started talking to each other, One was doing well, and the other one was dying. So, very difficult when you start to see that. But I think you have to, you're right, you got to hold your position and say, we're here for evidence-based medicine, however painful that is.
Judy.
David, it's not the main point of your talk, but I'd like to push back on the comment you made about now this drug is going to be generic, it's going off patent, and so the sponsor doesn't want to do a registration trial. They just want a publication and will change practice.
Right.
And I actually think that as academics, we really need to push back on that concept, because what you've got is a situation where it's not in their financial interest necessarily,
right.
But Danny's suggestion is a way to get around that and still have it done with the rigor that would be necessary to actually change practice and have it transparent enough that this data is being reviewed carefully and is all up front. So I just really worry when they try to kind of change practice kind of backdoor, and there are a lot of studies now showing the difference between what a protocol said, how many times the primary endpoint was changed during the conduct of the protocol, and what the ultimate publication said versus what the protocol actually said.
Right.
So it really makes me nervous if we just kind of accept that. It's a good way to do it. Just don't go through the trouble of an IND and a new.
So this will be a protocol-driven trial. Data will be collected by ACOSOG. So there's an independent data collection center separate from the company. So we will then eventually report the data out, and data safety monitoring committee and all the bells and whistles that go with it.
But so if we're going to want to change practice, why shouldn't that be submitted for a new indication to FDA?
I think the patent is going to expire. That's the problem. Okay.
That's a financial reason. That's not a scientific reason.
Right. So there's no interest in paying for an FDA registration when there's only 2 years left on the patent. So that's the problem.
And we have time for one more question. Perhaps I'll ask it. The oncology networks, and including ACOSOG, have been very, very successful over the years, whereas other disease networks have been less successful. So could you give us a feel for why these oncology networks have been as successful as they have been? Is it disease-specific, or is there something that you're doing that we should be paying more attention to?
There's a large infrastructure that's already in place that's funded by the National Cancer Institute. So, for example, the Z9001 GIST trial, 750 patients were enrolled into that trial. It's a rare disease, uncommon. And so there were 234 enrolling sites into this 750-patient trial. Mm-hmm. Now, no one in industry, no one here at the DCRI can run a trial like this because it's not cost effective, to have 234 enrolling sites. It would cost a fortune to do a trial like that. So Novartis gladly let one of the cooperative groups and the NCI underwrite some of that cost. And there is a huge membership database that's maintained by all the sites around the country by the NCI. It's a common database of membership and 1572s and things like that. They also provided that they distributed the drug for Novartis. I think Novartis paid the NCI some funds to do that. But they had a centralized pharmacy. So when you think about it, in oncology, there are a lot of drugs out there. There are a lot of sponsors, and they're all using the NCI as a means to distribute drug for rare diseases. Now, for Novartis, the imatinib trial, imatinib, Gleevec, it's a blockbuster drug for Novartis. The profit margin on this drug is close to $1 billion a year. And so, but they were not able to justify themselves to do a sponsored trial, industry-sponsored trial, back in 2001 when this trial got started, because they realized that they would have to find 234 sites, okay? And they can't do that. They can't afford it. And so there's a certain amount of infrastructure that's already in place that allows us to do rare disease. And you see that in pediatric oncology as well. And so unless the government underwrites some of this, we're not going to be able to do some of these trials, and we struggle. So actually, the GIST trial, this is incredibly rare in oncology, but the GIST trial we ran actually beat the Europeans in the trial. They had other adjuvant trials in GIST as well, and it's incredibly rare for the U.S. to beat the Europeans. But there they did not have the infrastructure that was in place here. So I think that helps a lot in how we do these trials, because 234 sites enrolling in for 750 patients, that none of you guys would do that with an industry sponsor. It would be cost prohibitive.
Well, David, thank you so much, and thank you all for joining us today.
Thank you. Pleasure.